We're racing against the microbe every day, and unfortunately, we're losing.
It's an arms race without end. The more drugs we launch at microbes, the more resistance they evolve. Maybe it's time to change our strategy. If we can drive microbes to evolve drug resistance, then we can also make them evolve in ways that benefit us. This is the radical proposition of Amherst biologist Paul Ewald.
When people are looking at the antibiotic resistance problem, they see evolution as sort of the bad guy. It's the evolutionary process that's led antibiotic resistance, and that's true. But just as easily, we can have evolution being the solution. In other words, we can have evolutionary processes leading to organisms becoming more mild.
Disease organisms evolve to be more or less harmful depending on how they are spread. Microbes that depend on close contact between people tend to be mild.
The rhinovirus that causes a common cold is transmitted by people walking around sneezing or coughing on other people. Since it really does depend on fairly healthy people to be transmitted, not surprisingly, it's one of the mildest viruses that we know about.
But microbes that are transmitted by insects or by tainted food or water tend to make people very sick.
The worst of all of the diarrheal bacteria that we know of have been waterborne. The bacteria that cause cholera and typhoid fever are often waterborne. So even if the organism is so harmful that the sick person can't move from bed, the organism can still be transmitted to large numbers of people. Once we understand the factors that favor increased harmfulness and decreased harmfulness, then we can look at all of the things we do in society. And we can ask the question, are we doing certain things, or can we do certain things that would favor organisms evolving towards mildness? We can look at the cholera outbreak in South America as a kind of natural experiment that allows us to evaluate these ideas.
In 1991, cholera invaded Peru and spread quickly. Over the next 5 years, more than 1 million people were stricken with diarrhea and vomiting, some severely. Over 10,000 people died. The disease was transmitted through water contaminated with human waste, or through food that was washed or handled by infected people. Ewald collected strains of cholera bacteria from South America and measured the amount of toxin they produced, an indication of their virulence. Over time, he would document evolution in action.
If you have contaminated water allowing transmission, we expect the cholera organism to evolve to a particularly high level of harmfulness, and that's exactly what we see. We find that bacteria that had invaded countries with poor water supplies evolved increased harmfulness over time. They've actually become more toxigenic. They produce more toxin than they did at the outset. If instead we clean up the water supplies, then we force the bacteria to be transmitted only by routes that require healthy people. And what we find is that when cholera invaded countries with clean water supplies, the organism dropped in its harmfulness. Those bacteria evolved lower levels of toxin production. They actually became more mild through time. People would still be getting infected, but the infections would be so mild that most people won't even be sick. So The cholera outbreak in Latin America suggests that we may need only a few years to change the cholera organism from one that would often kill people to one that hardly ever causes the disease. What we're suggesting here is that we can domesticate these disease organisms very much in the same way that we've domesticated other organisms that are potentially harmful. For example, wolves have been harmful to us throughout our evolutionary history, but through domestication, some wolves have evolved into dogs that instead of harming us, actually help us. And I think we can do the same thing with these disease organisms.
Working with evolution instead of against it, we might eventually subdue even the deadliest microbes. Evolution has already forged such surprising truces. Most wild cats have evolved a way to live with a virus closely related to one that is decimating humans. The story was unraveled by Stephen O'Brien, here at the National Zoo to examine a tranquilized cheetah.
Well, we originally became interested in the cats because I was interested in the interplay between infectious diseases and the genes of the species that suffer them. We began working with cheetahs and subsequently started to study each of the 37 different species in the cat family. What we're learning from them is that they are mirrors of evolutionary processes in humans.
It all began in the 1980s, when O'Brien became concerned that small populations of endangered cats were especially vulnerable to the ravages of infectious disease. Then he heard that domestic cats were falling prey to a newly discovered and lethal virus, the feline immunodeficiency virus, or FIV.
FIV is associated with very skinny and malnourished and wasting disease in house cats. And that disease was the result of the collapse of the immune system. So the parallels with human immunodeficiency virus were very strong. I was curious as to whether or not the virus had also been able to infect non-domestic cats.
O'Brien had collected biological specimens from thousands of wildcats around the world. He began to screen them for the presence of the virus.
Well, when we did that in cheetahs from East Africa and the pumas in the Rockies and the ocelots down in the Andes and the lions in the Serengeti, it turned out that virtually every species of cats on the planet had been exposed to and infected with a version of feline immunodeficiency virus. Well, I was terrified because I thought that we were just a heartbeat away from an epidemic that was going to decimate some of these cats. And since 36 of 37 of these cat species are already considered endangered or threatened, then this could be the final wallop.
For years, O'Brien feared the worst. He urged zookeepers and game wardens around the world to test their animals for the virus and to watch for AIDS-like symptoms.
What we discovered, though, over time, is that these cats were really not getting ill. It was as if they had somehow come up with a resistance to a fatal virus.
O'Brien's research suggests FIV first infected the cat's ancestors around 1 million years ago. It decimated the animals. But a few cats carried mutations that made them resistant to the virus. These survivors passed on their protective genes to their offspring and to most wildcats alive today. Over time, the virus may also have evolved into less lethal strains. Today, wildcats and FIV have reached the end of a long evolutionary process and have adapted to each other. humans and HIV only recently embarked on the path that might eventually lead to a truce. But the example of the wild cats convinced O'Brien there must be people endowed with mutations that protect them from HIV. He set out to find them.
Over a 10-year period of time, I quietly collected blood samples from 10,000 individuals that are high risk. My colleagues and I extracted the DNA, and we were stunned to discover a whopping mutation which protected against HIV infection. And it was the first gene that we could definitively say was influencing the outcome of exposure to this deadly virus.
Most people have receptors on their immune cells that allow HIV to dock and gain entry. But people with the mutation discovered by O'Brien lack some or all of these receptors. Infection by HIV becomes impossible. The mutation is present in about 10% of European Caucasians, but completely absent in native African and East Asian peoples. Something in the evolutionary history of Caucasians must have favored the survival of people with this mutation.
We've actually used precise dating techniques to date the last time such a selective pressure took place, and that came out 700 years ago. Well, if you look at the history books, that was the time of a rather dramatic infectious disease pandemic, which was the Black Death or the bubonic plague. And at that time, a third of Europeans were wiped out.
A mutation that saved people from the plague 7 centuries ago may now protect their descendants from infection by HIV.
Today, when we scroll through the genes of cats or humans, we discover that they're littered with these footprints of historic epidemics that have defined the survival of today's living species.
We all bear the marks of our ancestors' struggle for survival. But evolution is driven not just by conflict.
Okay, are there any questions about that? So if I was to draw a little graph here, and I had virulence on this axis, so how deadly it is, and so that's increasing. And then this is ease of transmission going from high or low to high. What do you think the relationship would look like? Would it be a line going up, a line going down? So if we have ease of transmission is low, so there's little, so it's hard, right? So we'll put hard, right? High, so this would be easy. So how would that relationship look? This or like that?
Are you talking about viruses that are not?
Yes, or the cholera, or the HIV.
So the cholera would go down.
Well, that tells me that it's going to be high virulence if it is hard to transmit. So it's actually the other way. So it looks like this. So here we have, it's hard to transmit, so there's low ease of transmission, right? And so virulence is also low. So that's a kind of a relationship between those. So you might predict that now that we've made it much harder for the HIV virus to be transmitted, because we now practice safe sex, right? And we don't allow for transfusions without testing the blood, and we give people clean needles, you would predict that over time, the HIV virus is actually going to become less virulent. And so people might be able to live maybe 10 years without ever getting sick, or they might actually be able to extend their lifespan and live with taking retroviral drugs and for a much longer period of time than they did in the past. Then the other idea is that there are some mutations that arise that actually make it impossible for the HIV virus to infect cells. And I was just thinking that I do not believe, like when you do a genetic test like 23andMe, I don't think they test for that, but that would be really interesting, right? So maybe they're testing for it, but they're just not disclosing it to you. Because all of those genetic companies, they're actually quite inexpensive. So you can get your genome tested for what, like $200, $100 special, right? But what they're hoping to do is do a big database of people so that they can then do research on that database and see correlation. So it'd be interesting to see how many people who are tested in 23andMe actually carry the protection against HIV, right? Might not be something that they would want to tell their customers though.
Yeah.
Like, oh, I don't, it's not a problem, right? So.
Oh, no, I would never get HIV.
Okay.
Practice
Yeah.
the same thing.
Yeah. Okay. So there are other immunodeficiencies besides the acquired one. So acquired means that you have to get it from an infection, from a virus, in most cases. But we also have genetic autoimmune deficiencies. And there is a one that has become very famous that is referred to as SID. And SID is the bubble baby disease. Right, or the bubble boy disease. So you don't need to know what SID stands for, but it's severe combined immunodeficiency. And so this is actually due to a mutation that you are born with, or that these babies are born with, where their lymphocytes cannot mature. So they have essentially no lymphocytes or no mature lymphocytes. So these are the children that they have to keep isolated. And so SID here is a child that actually lived quite a longer life than they thought he would live. So here he is actually in his house, and he's able to interact with the people around him, but he cannot be exposed to even the most minimal type of bacteria because his body is not able to fight it off. And so these children normally die quite young, but there is a new way to treat them, and that is through gene therapy.
Do they die of because they get sick or because their body just can't?
The problem is that there's a couple of things. We actually do need microorganisms in our body. So we need like good gut bacteria, right? And there's actually good bacteria on the surface of your skin. And so I think that probably they aren't healthy. Yeah, failure to thrive. They probably can't digest their food as well, right?
Because they're if they were exposed to it, it's not so weak that they can't adapt to it enough. Then it's just too much for them to adapt.
Right. So if you kept them completely isolated, they might be able to survive for longer. I don't know if there's any possibility of that. But, they've actually shown that you can take mice or rats and you can completely sterilize them and you can grow them in a sterile environment, and remove all the microorganisms from their body, and they are very sickly. So you have to kind of be exposed to microbes because their digestive system, especially for a rat, because they actually digest cellulose, so they need some bacteria, but they're not healthy. So sterilizing yourself and getting rid of all your microbes, you're going to be sick. Okay, so with gene therapy, what they're able to do is they've actually isolated one of the genes that causes it, and so they remove bone marrow cells. So these are the stem cells that give rise to T lymphocytes. They then inject them with the normal gene. They grow a colony of the cells, and then they put them back into the body. So this has been treated quite successfully with gene therapy. So this is just a diagram. One of the ways that they grow the gene, or they get the gene initially, is through bacteria. And then they actually put the bacteria into a virus, and then they let the virus actually infect the stem cells. And one of the things that viruses do is that they insert their own DNA into the host cell. And so this is the one where they've actually used the virus to get this new gene into the stem cells. And then they put them back into the bone marrow. It's like a bone marrow transplant. And then those cells are able to reproduce, and they are capable of maturing into the T and the B lymphocytes. Those babies would have innate immunity, they just don't have adaptive immunity, right? So they would have the macrophages and the phagocytes and all of the other stuff. It's just the adaptive immunity piece that they are missing. Okay, so another problem with the immune system is what is referred to as hypersensitivities. And this is where your immune system responds to substances that are not going to cause you harm. So I'll put inappropriately. Responds to substances. So it could be a food allergy, or it could be the venom from a bee sting. So these are typically referred to as allergens. And so these allergens could actually be multicellular. So for example, pollen, well, actually pollen is unicellular, but they could be multicellular. Let's see, what would be an example of a multicellular allergy? Oh, now I can't think of it. What would be an example? I'm sure we have some allergens that are multicellular. So it could be like dust mite feces. It could be, that's a really common one. Wheat pollen, toxins like bee stings. It could also be food allergies like peanut allergies, or even gluten, which is a type of protein that is found specifically in grains like wheat. And so what these allergens do is that they cause the production of IgE antibodies. So these IgE antibodies are produced when you're first exposed to the allergen. And so one of the really interesting things about allergies is that if you're kind of an allergic person, so you tend to have allergies, that can be genetic. But when you first move to a new location, it could be that you feel really good in that new environment initially. So it takes about 2 years of being sensitized to the allergens that are in this new environment before you actually start having the allergies. And so this is the first exposure to allergens. Okay, so the first set of the primary response is the first exposure. And then the Ig antibodies get inserted into the membrane of mast cells. And these mast cells are capable of producing histamine. So these produce histamine. So this would be the second exposure. or the secondary response. And so what happens is those antibodies that are in the membrane bind the antigen, and that causes the massive release of histamine, and it leads to inflammation, right? So this leads to inflammation. Now, oftentimes you don't realize you have been exposed to something previously, or maybe in some cases you might not have been exposed to something previously. So this is the main pathway upon which it occurs, but you could have a massive allergic response without even knowing that you have been previously infected. And so one of the problems with these massive allergic responses is that they cause a type of vascular shock called anaphylactic shock. And anaphylactic shock is where you have a low blood volume. And the reason for this is that the histamine has caused so much of the exudate to be formed as the fluid leaves and moves into the tissues. And it can also make it very difficult to breathe, right? So you get swelling of the mucous membranes, which makes breathing difficult. So this is why they recommend that if you are a parent of a young child, you always have antihistamines like Benadryl. So Benadryl works really fast, but the problem is that it crosses the blood-brain barrier, and it also makes you very sleepy, right? So they're the pink pills, the Benadryl. But they also, if you are getting a vaccination, or if you go to the doctor, they do have the ability to respond to anaphylactic shock. And so they use generally an EpiPen. So the EpiPen, and this was in the news, and it was very controversial because the people that were making the EpiPen all of a sudden increased the price like by 200%, right? And so it was like thousands of dollars to buy just a shot of epinephrine, right? And so people were like making their own EpiPens, right? Because it was ridiculous. And I actually believe that guy got put in jail, the guy that did that. I think he got in a lot of trouble for doing that. And so the EpiPen is epinephrine, which causes vasoconstriction. And this increases blood pressure. So what we want is to have a high enough blood pressure that you can get blood to the brain, right? We don't want to go into a coma. So the epinephrine does that. So it boosts the blood pressure. Now there can be other things that cause the release of histamine and swelling. And one of them really, which is really interesting, is you could actually have exercise-induced allergies. Right? And I actually only know this because I had to do research on this because I had this. It's crazy. So what happens is in the fall, when it starts to get cold, I start to wear pants and I start to walk to exercise, for example. And so when I walk, it is the movement of my legs and exercising that actually causes histamine to be released by the cells in my skin. So has anybody ever felt really, really itchy when they're outside exercising? Yeah. Yes, right. And so I thought that it was poor circulation. So I'm like, what is wrong with my body? Blood is pooling in my legs. My legs are turning bright red, and I just want to scratch them because they're so itchy, right? And so what I later learned that it doesn't have to do with circulation, but it has to do with the release of histamine. So for some reason, and my sisters have it in their arms. I don't have it in my arms. I have it in my legs. So for some reason, just the activity of exercising, specifically in the cold. So cold can induce this, but just the friction of like clothing on me in the cold causes the release of histamine. So now I take an antihistamine before I exercise. And if I go, it used to get so bad that I would actually have to sit down and put my legs up in the air and call my husband to come get me, because literally I could not walk. Like, it was so crazy. And so now I take a Benadryl with me if I exercise, and I take the Benadryl, and that causes it to just go away immediately.
It's almost like the feeling when your feet fall asleep.
It's hot.
Yeah.
So it's not pins, but it's hot, and you just want to scratch it, but scratching makes it much itchier, and it can be really, I mean, it's physically painful. It's really.
I found that I've gotten more.
Yes, I'm not sure why it's cold.
When I'm like,
Yes.
and it just,
Yes, I have no idea.
I got it pretty bad when I played basketball here for the college, and like my legs would just sweat. Like, you don't, I mean, you're inside, so you don't really realize that part of your body sweats so much. And when I looked down, sometimes my legs would sweat like people's heads do. And that's when it was really bad. It was almost like it was sweating out and starting to irritate my skin.
Right.
Because I went to the trainer for it. I was like, my legs are always on fire. Like, it started to kind of happen in my muscles too. And you need to drink more water. Like, I just was, you know, I wasn't dehydrated, but because I was lower dehydration, that stuff was
affecting it.
Mm-hmm.
Or maybe your sweat was making you itch, because sweat can be very acidic,
Yeah.
and so it can actually burn your skin if you have very acidic sweat. Okay. So the last thing we need to talk about as it relates to the immune system, actually first, I'll just note that this is a diagram that shows from your new textbook the different types of hypersensitivities. So we were talking specifically about allergens that are binding to mast cells. And so mast cells have granules of histamine that they just store. And once the allergen binds to that antibody, the granules are released, and that is called degranulation. And then there's other types of hypersensitivities that are associated with different types of diseases. So, for example, this is mismatched blood. So your transfused blood. This one is if you have diabetes mellitus, like type 1. This is where your, for example, it can actually cause a hypersensitivity. That is where we're going to talk about. This is autoimmune disease. Okay. So autoimmunity is actually another type of hypersensitivity. And make sure that you realize that this is not acquired. People get that confused all the time on quizzes and exams. So it's different from acquired. It's auto. And this is where your immune system fails to tell self from non-self, right? And so this is where the immune system attacks normal, healthy tissue. So some examples of this that you might have heard of previously include like rheumatoid arthritis, which is what is shown in the diagram. So this causes inflammation in the joints, and the inflammation is so severe. This is what the immune system is doing to the body. And so it's actually kind of causing the bones to become deformed. So people that have like big joints, like knuckle joints on their fingers, and then they sometimes lose the ability to use their hands. That would be an example of rheumatoid arthritis. We also have one that is more systemic, like lupus. One of the characteristics of lupus is that they get a butterfly-shaped discoloration on their face. That's the kind of the unique characteristics that's called the butterfly rash on the face. And the lupus causes a more systemic, so this would be an example where they have a low-grade fever, they have inflammation around the heart, they have inflammation in the muscles, they tend to get ulcers in their mouth, and they tend to lose their teeth. And so lupus is just the immune system attacking what would be normal, healthy tissue. So we could ask, why does this happen? And there's a couple of theories behind it. One is that the immune system could actually be influenced by a virus. So you could have a viral infection that then causes the immune system to kind of go crazy, and it will stop learning to detect self from non-self. So some immune or autoimmunities, some are due to a previous viral, or can be related to a previous viral infection. So this viral infection somehow caused the immune system to become hyperactive. The other interesting thing about this autoimmune disease is that 70% of all people that get autoimmune diseases are female. So the question is, why do females get it so much more frequently than other individuals? And so there was a hypothesis that was put forward that females are not just their own cells, but if they have become pregnant, they actually could have fetal cells that are found in their tissues that could actually be causing the immune system to attack those tissues. And so the idea behind that is what is related to a phenomena that is called microchimerism. So if we look at what a chimera is, a chimera, and there are actually human chimeras, a chimera is a person who has two different types of cell lineages in their body. And this can actually lead to autoimmune diseases as well, because this is oftentimes due to twins merging together. And so they call this the missing twin phenomena. Yep. You're a chimera?
Well, I guess when my mom went in the first time, well, so she found out she was pregnant at home, went in, had her first ultrasound. They said that they found 2 heartbeats. So when she went in for her next one, there was only 1 heartbeat.
Oh, so interesting.
Hey, my twin in the womb, I guess.
I know another chimera in Pendleton, but she really does have health problems. And so she went to OHSU, and so she got diagnosed with it.
No.
So it really does affect her
I
health.
just like to play like I did. I don't know. I was very early in the pregnancy to where I bit my mom.
I had another student who said that she was born with a growth on her head. And then when they cut it off and they analyzed it, it was her twin. Yeah. Yeah. Okay. So this is where 2 embryos fuse. Right. So you have 2 embryos. So they could be, we'll assume that they're not identical twins. It'd be more likely that they would be fraternal twins. So they would be have 2 different lineages. And I guess if they were identical twins, you wouldn't be able to tell, and it wouldn't be that important for the immune system. But some people are born where they might have male cells forming their liver, and then the rest of their body are all female cells. And so that liver was derived from the embryonic stem cells from the twin. But the problem is that a person's immune system is going to see that liver as not being self. And so they might attack it. And so this is where you could get the immune system attacking tissues that are of the wrong cell line, right? So that's the chimera. So the micro part means that there isn't a lot of cells, there just are a few cells. And so this can be due to, so micro means that there are a few cells, and they can be derived from fetal cells that cross the placenta in a pregnant female. So they cross the placenta and take up residence in the mother. So this, we thought at one point that the placenta was like this wall and nothing could get through it. Now we realize that there are antibodies moving through it. There are viruses like the HIV virus moving through it. And now we know that cells can move through it. So fetal cells can go from the mother, and this actually could be a kind of a good thing because fetal cells are stem cells, and so they could actually go out into the body and repair damage. So it's really interesting that a female that might have been pregnant 3 times in her life could actually have 3 different cell lines in her body, including her own. Okay. So it was thought that males, so this was kind of a hypothesis about autoimmune diseases in females, but we know now that the cells go the other way. And so now we know that maternal cells do cross into the fetus. Okay. So you might, if you're a male, you would have no chance of being a microchimera. But in this particular instance, you would. So if you're a male, you could have some of your mother cells in your body. And if they are detected as being foreign, that could be a source of autoimmunity, right? Because it's not really, the immune system is actually detecting them rightly as being non-self, but it is a bad call because the maternal cells are obviously not going to do damage to your body. And so, that's kind of interesting. And so there was recently a lot of newspaper articles about chimeras. And so this is this idea that when we're sequencing DNA now, we're finding all kinds of really interesting things. So if you're a male and they sequence your DNA and they find female cells, female genes, genotypes, that could be the source of it. So microchimeras is kind of the new big area of research when they're talking about genetics and autoimmunity.
What's the odds that happens? Like, is it?
We don't know, but it's just because everybody's sequencing their DNA, we're finding it's more common than they thought it would be. Okay. So you are probably a microchimera, even if you didn't have a twin in your vicinity.
I don't know where.
Interesting.
I mean, I've been a really healthy kid my whole life. I mean, of course, I've had my sick.
Maybe it was an identical twin, because an identical twin wouldn't cause. But maybe it was so early on that the cells, that you don't have a lot of their cells in your body.
It's weird, too, because it skips generations. So I'm actually supposed to have twins. My mom wasn't. So when she found out she was pregnant with twins, she was really like, this is weird. So it was, but I've never even broken a bone. I've never, knock on wood, I've never been to the emergency room. I've been a really healthy kid. So
right.
Hopefully there's no weird stuff going on there. Hopefully I didn't actually eat my twins.
Absorb them.
So yeah.
Absorbed. No, you just came together. You didn't eat
one.
them. You became one.
Maybe I have another life.
Okay. Respiration. So this is the next big topic. And so when we're talking about respiration, we can talk about external and internal. So from the atmosphere, I could spell atmosphere. We have an exchange with the capillaries in the lungs. So if we look at those arrows, those show the movement of oxygen and carbon dioxide, which are needed for cellular respiration. Oxygen is needed, and carbon dioxide is a byproduct of it. So oxygen moves in, and carbon dioxide moves out. That should be one way. So they refer to this first part as external respiration. And the second part as internal respiration. External respiration has to do with ventilating the respiratory surface. Right? So we have to move air in to get into the respiratory surface. And there's lots of things conducting, we have to conduct it down into the inside. And the reason why we do this, why we don't have just our gills like in our neck, right? We don't have the gills in our neck like fish do, is that our respiratory surface is buried to prevent dehydration, right? So this is deep in the body to prevent water loss. All terrestrial organisms have lungs as opposed to gills. And that has to do with preventing water loss, because water would simply evaporate off the surface if we had gills exposed to the air. So internal respiration has to do with the diffusion of gases. And so we have to have a specialized membrane over which oxygen can just automatically move from a higher concentration to a lower concentration. So this diffusion is passive. It does not take energy. And gases will diffuse from a higher concentration to a lower concentration. So our body is designed to optimize diffusion. So we don't have to use energy to pump the gases. And so blood coming in to the respiratory surface tends to be low oxygen. That would make sense because it wants to draw the oxygen out of the air and into the blood. Right? And then blood coming into the lungs is also high in CO2. So CO2 will just diffuse from the higher concentration to the lower concentration and be breathed out. Okay. So what that means is that most of our respiratory system that we think of is actually the conducting zone, where it just is conducting air to the respiratory surface. So we have the conducting zones. And these carry air to the respiratory surface. Now, our respiratory surface is actually a structure called an alveoli. And the structure of the lungs allows for a very large surface area over which diffusion can take place. And so the respiratory zone is what is referred to as the alveoli. Okay, so that is where gases actually diffuse. The rest is just conducting. So gas doesn't move across the trachea. Gas doesn't move across the nasal cavity. It's only being conducted to the alveoli, which is where the respiratory surface is. So something interesting about our conducting zone is that it is separated from the mouth. So we actually have a hard and soft palate here, which separates the nasal cavity from the oral cavity. When you look at other vertebrates like amphibians, they lack that. And what that means is that they just their nasal cavity just delivers air directly to their mouth, which makes it hard for them to chew and breathe at the same time. But we can chew and breathe pretty well at the same time. However, one of the problems is that sometimes food can actually get into the respiratory system and then, or block the respiratory system, and then we can choke and we can die that way. And so it's really interesting that our respiratory and our digestive systems are so interconnected, because if you think about other organisms like, say, insects, they have a mouth that they get food in, and then they have holes in their abdomen in which they breathe. And there's no problem with ever getting food clogging up your respiratory tract. Okay, so it's kind of an interesting design, and there is one problem of that. Okay, so we're just going to talk today about the nasal cavity. The opening to the nasal cavity are the nares, right? So these are the nostrils. And they open up into what is referred to as the vestibule. Now, the vestibule is that place where you can stick your finger. Okay, it's not really high up in there, and it is really protected. So it is actually stratified squamous epithelial, right? So it's not going to easily bleed due to, putting your finger up there, right? So this is stratified squamous, not with an E, squamous epithelial tissue. Okay. It also has sweat glands. And the sweat glands are actually designed to humidify the air. There's also hairs. So there's nose hairs. And these nose hairs are actually designed to capture large debris. So when you're out working in a very dusty environment, you might notice that you have to actually go home and clean your vestibule out, right? Because it gets full of stuff, full of junk. And that is protective, because one of the functions of the nasal cavity is to filter stuff out of the air so that it does not get into the lungs. And that's why people always say it's better to breathe through your nose than it is through your mouth. Because if I'm breathing in through my mouth into my lungs, then I'm going to start to get dust and other things in there, and it's going to take a lot of work by the cells to get that out of there. We don't want dust and debris to get directly into the lungs. So there's an interesting image, not in your textbook, but in the other textbook, that actually shows a cadaver. So this is a sagittal, a mid-sagittal cut through your head. And so this right here is what is referred to as the vestibule. And then once you get beyond the vestibule, this is not something that you can easily reach with your finger. And so this is what is referred to as the respiratory surface of the nasal cavity. And it is actually not stratified, and so it will actually easily bleed and can be easily damaged. And so sometimes when you breathe in really hot air and get dehydrated, then that tissue will dry out and it will start to bleed, and then you'll get nosebleeds, right? And so we have see if they have it listed here. No, they don't. Let's look at this diagram. So this is a diagram from the new textbook. And so you see that we have the rest of the nasal cavity. And so this includes, so this would still be under nasal cavity, what is called the olfactory epithelium. So when you hear the word olfactory, what do you think of? Smell. So these detect and bind airborne chemicals. So chemicals that are in the air, which could include scents from food that as you're chewing, some of that food actually becomes, the scents of it become airborne and then go up into the nasal cavity and you can smell while you're eating. So these are really interesting because they extend down from the superior region. So this is the top of the main part of the nasal cavity. And these olfactory nerves just kind of dangle down inside the space. And you'll notice also that that is the roof of your skull. So there's actually holes that are referred to as the cribriform plate in the ethmoid bone. There's holes through which those nerves travel and send impulses to your olfactory bulb. And so if we look at this olfactory epithelium in the cadaver, you can actually see it. This is so cool. So this would be where your olfactory bulb would sit in your brain. This is the top of your skull. And so you can actually see these nerves and their cytoplasmic extensions kind of dangling down into the nasal cavity. One of the things about smell is that these are actually replaced quite frequently. So if you damage them by breathing in a really bad chemical like ammonia can really affect your smell. You can damage the nerves, but they tend to regenerate and replace themselves. So you generally will regain your sense of smell. Okay, so I'm going to stop there for today, and then we'll continue on with our discussion of the respiratory system. And